Oliver, take
Yeah,
it away.
thank you. Great. Yeah, thanks for the introduction, Archie. And yeah, let me just share screen and start with a bit of slides and background material on for everyone, so we can level set. We know who's going to be speaking, and yeah, well, then we can move on to talk a bit about DeepChain, which is InstaDeep's protein AI guided protein design platform. So today, yeah, we're going to talk about protein design. We'll try and give you a bit of a flavor in healthcare, but of course, protein design is generic across many industries, but healthcare should be interested in this. And yeah, we see this as a novel branch of machine learning with high potential for impact on the continent as well, not just in other markets where we work at the moment. So to give you a bit of a quick intro of the people presenting today, there's going to be me. I'm a senior software engineer at InstaDeep. I did my PhD in machine learning at the University of Oxford. I actually spent 7 years working in the industrial research in Nairobi, Kenya. So I'm quite used to working on direct challenges on the continent. And I will let my colleagues talk about themselves when they take over. But for now, I'm going to give you an intro into a bit of the background on proteins, and then jump into DeepChain. And then Bakri will talk a bit about code and interacting with proteins in a code-based example in our notebook, and how it will extend these machine learning approaches on top of it. So yeah, let's get started with what is a protein for everyone joining. So as we don't know the background for everyone, apologies if this is too basic, but also is something that I didn't remember so much from high school when I started working on protein design again in machine learning. So proteins are large molecules made up of amino acid chain sequences or residues. There are 20 different naturally occurring amino acids, and these and proteins are manufactured inside cells. Let's say inside human cells, predominantly for healthcare applications that we're interested in. And DNA is transcripted into mRNA, which is itself translated into a protein. There are about 189 million sequences recorded in the UniProt database. Of course, there are orders of magnitude more naturally occurring proteins, but these are ones that have been studied, and 193,455 proteins in the protein data bank. That could have changed since this was written, but these are structures that have been mapped and studied through crystallography. Yeah, so these amino acid building blocks of proteins are organic molecules that contain a backbone, which is common to all amino acids, and a specific side chain residue that defines the function of, or defines the structure and the function of that amino acid as it's built up into a protein. So there are 20 types of natural amino acids, as I said, and each of these we give a letter-based index, 20 being less than at least the alphabet that we use most commonly. And not all of these natural amino acids can be produced by the human body, but some might be brought by food. I think that's somewhat tangential to the discussion, but it's just interesting to know where these amino acids come from to start with. So proteins in healthcare, why would we care so much about proteins in healthcare? So there are about 1 to 3 billion proteins inside you at any time, 50 million in a single cell yeast, or 2 million in a bacteria that define them. So our bodies are around 15% protein, and proteins are responsible. They're kind of these biological machines in our bodies. They're required for nearly every task of cellular life. So whether that's regulation of things like blood sugars with insulin, transport of oxygen with hemoglobin, protection of our organisms through our immune system and antibodies, or providing structure to our organism with things like collagen. Any single change or mutation in an amino acid can directly affect the protein's behavior. So one replacement or removal or change of an amino acid in a protein that could consist of thousands of amino acids could directly change its function or how it performs the task that it does, making it potentially unstable or improving its stability, affecting its folding, charge, and activity, among other things. So really affecting the task it was designed to do. So we could think of protein structures to be represented by some primary structure. If we go back to this notion of it being a chain of amino acids, a protein can be represented by a graph, a skeletal formula that gives simplified information about atoms and bonds between the elements, essentially, in the protein or the amino acid sequence. Each amino acid has a backbone that's common to all amino acid types, and a side chain which is specific and defines that amino acid. So apart from the hydrogens, which we're not showing linked on this carbon backbone here, it comprises one oxygen atom, one nitrogen atom, and two carbon atoms, C alpha and a carboxylic carbon here bonded to the oxygen. The first atom of the side chain of the amino acid is always might be referred to as the beta carbon, except for glycine, where you can see that actually this smallest amino acid doesn't really have a functional group. So here we've got a section of amino acids chained together to make this polypeptide, built from 5 amino acid residues. And at a local scale, in these chains, the three-dimensional shape of an amino acid sequence is parameterized by a set of bond lengths and atoms, and bond lengths and angles between the atoms. The bond lengths are mostly determined by the 2 atoms involved in the covalent bonds that form the structure, and they exhibit small variability. So this could be in the order of 1 or 2 angstroms, or an angstrom being 10 to the minus 10 meters. Bond angles are then the angles between 3 atoms within a plane. So we can define these dihedral or torsion angles around a bond, and angles between the atoms, between 2 planes. Now, just sharing this to give you a notion that this structure of an amino acid building up to a protein is a 3-dimensional problem defined in 3 dimensions. So moving from the primary structure to some secondary structure of a protein, we can now look at local 3-dimensional structure, where the 2 common forms of secondary structure are alpha helixes and beta sheets, which is mostly determined by the existence of hydrogen bond interactions. So just to add on that here, you can see a bit more clearly across the backbone of the amino acids how these bonds might form to give these secondary structures through hydrogen bonds. And on top of these secondary structures, we start to build tertiary structure through things like disulfide bridges, ionic bonding here, along with different hydrophobic interactions or induced van der Waals interactions or further hydrogen bonds that determine a tertiary structure or folding, and then we can introduce more partners to get further structural behavior as well, which we'd call the quaternary structure. So if I just seed this notion of structure prediction now, we can go from an input sequence of amino acids, so just a string of letters, or in this case, we've given 3-letter codes for the amino acids, and often we don't actually know what this sequence would look like, so we would need to predict the 3D structure just based on the amino acid sequence. And we're going to talk through further about how we might score that. There are some functions in DeepChain that do it. Likely you've heard about systems like AlphaFold, which perform this, and also how you might be interested in small perturbations in 3D structure for design tasks that improve some function through mutations. I'm just gonna make sure that people saw my message, because at this point, I'm going to talk a bit about DeepChain just in slides, and then we're going to move to the platform. So if everyone who doesn't already have an account could just go to deepchain.bio and sign up. It's a free account. You just need an email address. And yeah, thanks Archie. And we'll move into a practical example there, just to give an overview while people are doing that, or if you've already done it. There are kind of three main features we might be interested in for now. We're actually going to focus on this playground feature due to the nature of the licensing we do on DeepChain at the moment. Actually, the features of structure prediction and mutation evaluation and AI design are still quite computationally heavy tasks. So access to them requires a small license fee just to make sure that we can put some limit on them and aren't running a lot of GPU hours with users running lots of experiments. Structure prediction, I guess we seeded the idea earlier, but it allows us to use the DeepChain platform for anyone interested in protein-protein interaction to run what we're calling end-to-end in silico experiments. So why is something like DeepChain so interesting? For me, especially on the continent, I mean, everything is run in silico. So we don't have to have this extensive investment in wet lab biology or equipment that would allow you to do traditional protein design potentially. You can start to look at challenges immediately using compute, which is orders of magnitude less costly, and applying that to problems of interest that might not necessarily have the focus of previous labs. And yeah, I think this is a really interesting opportunity with features like the playground, where you can develop machine learning apps on top of protein design and run those directly in DeepChain, or run them in your code, as we're going to show you, to again continue to explore problems of interest, designs of interest that could directly impact the largest disease burdens on the continent, and bring insight into diseases like COVID-19, malaria, pneumonia have been discussed, or even tackling non-communicable diseases like diabetes. So yeah, structure prediction is a feature that perhaps I'll be able to demo for you, but unfortunately you won't be able to run directly. But starting with your amino acid sequences, we can fold those together using our machine learning models to go from just a language to of this amino acid sequence to a 3D folded structure, to then perform further experiments on top of that. Perhaps you want to mutate some of the amino acid positions and see how that influences its form and function. The DeepChain playground lets you create your own machine learning tools or apps to solve particular biological questions. So here we hope to, in what we talk about more extensively, democratize AI-based protein exploration and design by allowing students, independent researchers, people interested in healthcare to tap into our data sets, platform, pre-trained models, and compute power behind this to solve novel problems. And we give a platform for people to evaluate mutations and do AI-based protein design, so exploring multiple mutations at a protein interface by running physics-based simulations and machine learning scorers to guide protein interactions that may be improved. So we may improve the binding or disrupt the binding in some interface. And I'll go through a very specific example that we have documentation behind, so I don't lose too many people, and let's jump into something interactive where you're doing it yourself and it's not just me talking, and you get to feel for this. Great. So maybe everyone on the call, if you just put a yes or no, if you're able to create a DeepChain .bio account while I load up the page as well. I think through the Indaba, InstaDeep's had quite a lot of visibility, but you can also reach out on these platforms, but I won't spend any more time here. Let's jump to DeepChain. Great. So I've not seen in the chat any yeses or nos. I'm gonna go and log in because I already have an account, but if you haven't created an account, please go to sign up. Unfortunately, it's going to log me in directly because it's got my credentials stored. So when you log into DeepChain, what you're going to get is actually this design to start with, and we have a nice visualization of the quaternary structure of 4 chains here for a particular protein. Now, sorry, I do just need feedback from people on the call if you're at the same page as me and can follow on.
I'm actually just signing up right now.
Okay, great.
Maybe give us like 2 minutes or something.
Yeah, let me pause for a couple of minutes while people just drop in the chat that they were able to sign up at DeepChain.bio.
Right, I'm in.
You're in. Okay, we'll use you as the benchmark for how long these are
Cool.
taking. So great. What we're going to do is we're going to go through a design ourselves, actually. So we're going to use this structure, which we'll talk about a bit more in code as well. But what you can do in DeepChain is you can directly pull in a protein structure from a database called the Protein Data Bank. And all you need to do is give this 4-letter Protein Data Bank ID. So that's called the name of our design, AML workshop or something like this. Obviously, not very descriptive of the protein's actual form or function. Rather than the default ID, you're gonna type in 3MZG as the PDB ID. So this is the 4-letter PDB ID, and we can create a design around this structure. And so what 3NZG is, it's a two protein partner of prolactin and well, we'll get the name of it. The number of protein for the free version is limited to 1. Okay, well, I've already done this, so you will be able to include 3NZG in your project, and this is what you'll get. So I'm also using the same license as you. I'm not using a special internal InstaDeep license, just to make sure that we're all using the same thing. So we've got the prolactin, which is a hormone in the production of breast milk, for example, in human females. And we've got the prolactin receptor, which is, so here we've got the prolactin hormone itself, chain A in green. And depending how you are on colors, this looks a bit orange to me, but I guess it should be red. We've got the prolactin receptor here, which is the chain B. You can see the sequences for these two proteins in the partnership, and yeah, we can just in this viewer play around with different ways to visualize it, but I think we'll talk about that a bit more in code later. I just wanted people to have the chance to load 3NZG into DeepChain, and we're not going to stick in the AI designer, but we're going to jump to the playground with the structure and pick the prolactin receptor structure. So this is the B chain, and the one we're actually interested in where mutations happen, because the prolactin receptor regulates the amount of prolactin in the body. And as background, and we'll talk about this in the code a bit more, we know there is a mutation at position 188 in the sequence. So this is position, this histidine at 188. We know there's a reported pathology around this position. If we have a mutation, so this amino acid changes to something else, it can disrupt the binding with prolactin, a lot, the prolactin receptor, and lead to a pathology. So what we have here is the sequence, and what's happened when we picked this sequence is we've run it through our machine learning language models, and what it's trained to do is now predict the most likely mutation in this position. So what you can see here is a heat map across the 20 amino acids, the naturally occurring ones that could occur in this across all of the positions in the amino acid. And this is a language model trained on a large corpus of proteins and working much in the same way as natural language processing might do to try and predict a missing letter or a missing word in a sentence. We can train our models on the language of biology or proteins to understand what given the corpus of naturally occurring proteins could be a mutation that occurs in the wild. So this 188 position, we already know that from a study, there's a mutation that leads to disrupting the binding between prolactin and the prolactin receptor that leads to pathology. Now we've used DeepChain to rank the top 10 sequences, the most likely transitions. And actually, spoilers here, the study is based on the arginine mutation, and this is this R. So these are only the top 5 most likely mutation given our model, our language models. We'll give you more information in the code later, but this top 5 mutation leads to the disruption in binding energy, which we'll simulate later. For the purposes of DeepChain, it is now you can see we have direct access to running different predictions. Again, we could look at prolactin itself as the hormone and understand what happens there. But I just wanted to show loading a PDB into the interface and running this machine learning model for free in the platform to understand what might be the most likely mutations in the sequence. And now we're going to go deeper into analyzing the structure of 3-MZG and ZG, so prolactin and the prolactin receptor, and analyze a bit more this mutation in 3 dimensions, as we talked about, as well as how we generate these language model probabilities of mutation. So with that, unless Bakary, Halil, Seydu, you feel I've missed out on something glaring, we're going to jump to Bakary in the notebook. And Bakary, I can let you share screen, or would you like me to share the notebook?
Yeah, I can share my screen. Yeah, I can do that.
Sure. Okay, great. So I'll stop sharing my side. I've not run over time too much. We're trying to be quick, and
Don't stress about time. We're still good with time.
I will. Archie, do you give access to Bakary to share screen, or is it me?
I did give him access.
Okay, great. You've
Okay,
got
let me
access,
try to
Bakary.
share my screen.
And while you're doing your share screen, Bakary, I can send out the link to the collab to everyone on the.
Okay. I think you should be able to see my screen now. If everything is okay.
Yep, all good.
Cool. Just to introduce myself, so my name is Bakary, and I'm a computational biologist working for InstaDeep. And so we're gonna just walk through this notebook and explore this protein that Oliver was talking about, the 3-MZG. So if you have any question, please leave that in the chat, or you can even stop me and ask any question at some point. So it should be pretty straightforward, and there is no much thing to change unless you are interested in a specific in any other protein, and okay, so a protein, a mutation in that protein, and you want to see how things behave when you mutate that position in that protein. So that might be interesting, and you can actually change things here to do that mutation on that protein. So, I mean, just we're gonna go and download the structure, visualize roughly, and look at some specific mutation position inside that structure, and then you're gonna mutate. And the interesting part is that you're gonna compare like before and after mutation, what happened, and what that mutation would cause to the structure. So that's going to be our analytic part.
Unfortunately, Bakary, I didn't anticipate sharing in Colab on our Google accounts is linked to only in-company sharing. So I've just been trying to publish it. I don't know if Halil, you can also look at the sharing at the same time as me. But yeah, while you're talking about this, unfortunately, those on the call aren't going to be able to view if I just give a link. So I'm just looking at republishing a version that they can have access to. But please keep going. Halil, if you can also look at republishing it at the same time, we'll get it sent out on the chat.
Yeah, so I can go through the notebook, like run everything once and comment on it, and then yeah, feel free to stop me at any point. So as Oliver was, hopefully you see my screen at least. Let me zoom in. Is that better?
That's much better. That's much better.
Okay, cool. So what we will do, so first we're gonna, I mean, if you have access to the notebook, we will install some dependencies. I think I already did that. That should go fast, and hopefully there's gonna be any problem, because as they say, never do a live demo. But basically, I mean, all we have to do is really go through this and run the notebook, the play buttons. So I'm gonna comment on the important part, but we will install some things and load some dependencies. And yeah, so we will start here. And then if you want to change anything in the notebook when you get access to it, so please just search for this string inside the notebook. It should indicate you places that you can change, like the PDB code that you want to look at, and the residue, the position that you are interested in the PDB. The position will be not sequence space, but the one inside the PDB, the residue numbering inside the PDB. Also, I don't know if you guys are familiar with proteins and PDB and all of that. So
I think yes and no, Bakary. Just yeah, just let's assume not. No. So yeah.
Okay. So
as much background.
I mean, there is a repository of protein structures. I think Oliver went to why protein are important and a lot of stuff. And then you, from that repository, you can pull any protein that you're interested, not any, but any protein that has a structure in that repository. So you can get that protein and analyze the structure. So the key thing is that the structure of the protein will always be linked to its function. So by looking at the structure and analyzing and doing some modeling on it, you can have a good glimpse inside the functionality of that structure. And so what we will do, basically we just download, so the website is called rcsb.org. So we just pass in this structure ID, which is the one of the prolactin with its receptor. So we run this, and basically I think the structure should always already be present. So it's here. Then let's just see, I think it's already there. And we load that to see visually how it will be looking. So you have, I think, the red part, to be honest, I'm not 100% sure, but the red part and the blue part, so there are two chains on the structure. So one is chain B, one is chain A, and one is the prolactin and the other one is its receptor. So this is just a graphical representation of the atomic coordinates. So you have inside the file, you have coordinate in 3 dimensions. So you have XYZ coordinate for the atoms. So as you can see, as I'm hovering around the structure, you see atoms and you see histidine 49. So right now I'm on this histidine residue inside the protein and on the C alpha. So this is chain B, histidine C alpha. So I can, I mean, it's just help to get a glimpse into how the overall structure is folded. And when you do that, you might be, most likely you will be interested in looking at specific regions on the structure. So here what we will do is that we, as Olivia was saying, there is a mutation inside the structure. So there is a change in the amino acid, which is causing some instability inside the protein. So we will be mostly interested in that specific position, and we want to see why that change in that residue is causing that instability inside the protein. So here we will be looking at position 188 in chain B. So again, if you are loading another structure that you're interested in, you might want to change this. It will take you to visualizing that specific position, and we will see the residue that is there. So I'm gonna zoom in here. So you can see here the residue. So this is the histidine 188. And also we have around it some other residues. And most importantly, it's bound. So if I scroll up, so you have this chain B here, and unfortunately we don't have that chain B visualized here, but you would understand that this residue is in close contact with this other chain, the one in red here. So anything bad happening to that residue might cause some destabilization of the protein, leading to losing interactions. So that's why we are interested in the mutation here. So just to comment, the histidine is this residue. So the balls that you see are atoms, and the sticks are bonds between the atoms. And so this is there is another representation here, which for the surrounding residue. So we have different residues. We have the main one here, the histidine, and around it you will have other residues. And the red dots, so those are the water molecules. So in the protein, I mean, in your body, like inside, you have like protein living in water environment. So you will have this kind of thing in almost all PDBs. I don't know if you are, if there is any question so far.
Yeah, just check, Bakri, you had one question from, okay, sorry. Abraham, I didn't see Halil that you'd posted. So you had two versions of the collab, just while Bakri answers questions, but in the chat there are two versions. And you had one question from Abraham, what about the bond types? Can they be visualized too?
What are they?
The bond types between atoms.
The bond type. So that's super good question. So right now, the bond, these are all covalent bonds, and that's the, I mean, the kind of interactions are really interesting. So this every time you see a stick, it's a covalent bond. So if most of the time the non-covalent bond, they won't be displayed in this visualization, but you can, I mean, you can play around with the different representations that you want, and then you can probably change it to see different non-covalent bond type here. Hope that answers your question.
Yeah, so for example, Husmedadin said there are several disulfide bonds in proteins, as we said in the presentation. Are these bonds represented in any other way other than the covalent ones? Maybe we don't have an example in this.
If we have a protein in which there are those kind of interaction, they will be presented in the same way, in this sticky way. So they will be connecting to atoms.
Great. I think you can keep going. I hope between Halil and my links for similar things, you have access to the code, whether you choose to run it now or want to just listen and maybe access it later.
Okay, hopefully this is clear that we're gonna see when we mutate that residue, what are the impacts on the structure. So here, what we will do is that we will look at the residues close to that specific histidine. So I'm gonna run this. You don't have to care about much what it is. So it's gonna find all residues close to that histidine. I need to change this to be, this residue index should be 1828. Let me just check that quickly. Okay, so this should be chain B. Okay, so these are the residues around the histidine in chain B. So you have the residue name, its residue index, and the chain on which it is. So so you can also define a radius. So here it's a radius of 4. So it's gonna go, and anything around that residue in a radius of 4 angstrom, it's gonna collect that and put that here. And you have the water molecules and you have the residues inside. And then now we're gonna do the interesting bit, which is to do the mutation on the structure. And to do that, you need to define your mutation. Again, if you are loading any other structure, you might need to change this here. So you will have to define the position, which is basically the chain and the residue number. And you want to define a target residue. So you want to define, okay, you can go through this Wikipedia paper, which explain a little bit more about the amino acids. So basically, commonly there are 20, and you can mutate to any of the 20 amino acids. So there is, and we can have a brief look to that. So I think there is a nice diagram of all the amino acids here. Yeah, this one. So they have different structure. And so this is the one inside the structure right now. So this is the histidine, and this is the arginine inside the structure. So usually you will have this common amino acids inside the protein. And then if you mutate toward one, or you might be disrupting the functionality of the protein. And here we are mutating to arginine, and we will see what is going to happen when we do that. So basically we use tools to do those kind of things. And here we are basically using PyMol. So it's a modeling tool. And so let's have a look inside. So what here, so this is the mutate function. So it's going to take our molecule, which is our protein, our input protein, the chain, the residue number, this is the chain, and then the target residue. So this function is really to create this mutated protein, which gonna contain, instead of the histidine, it's gonna contain the arginine. So here I'm just showing that if I grab inside that protein the coordinates, so this is the protein we loaded, the 3-MZG, and at position 188 we have the histidine. So if we apply the mutate function, so we're gonna input our protein and the chain and the residue index and the target residue. So this should be the arginine, yeah. And we run this. So I'm behind the scenes. So what's happening is that these residues, they have different rotamers, and they have, so what the mutate will select the best rotamer in that position, basically, for this arginine residue. So you have a summary of what happened, basically. So it selected the rotamer number 19 among the arginine ones. And now let's look again inside that structure. So right now we are grabbing on the mutated structure, and we want to see what happens. So now we have different atoms, and these are all from the arginine. So instead of the histidine now at position 188, we have the arginine residue, which is now at that position. Now this is, I mean, we will visualize it here, but behind the scene, this is all that is happening. So you have some atoms with some coordinates, and those atoms and their coordinates, so you will have a change of that. And as a result, you will have a change also in the environment, the chemical interactions, and all the bad things that could be, all the bad things that can happen. Any question? I don't know if I'm going too fast or.
There were a few more added questions on the understanding the bonds. I don't know if we've moved on too quickly.
Because I don't even see the chat.
Yeah, sorry, in the chat screen. So yeah, it was on the importance of bonds on the final fold or structure of the protein. Should we infer that non-covalent bonds, for example, hydrogen bonds, have less impact on the result? Well,
sorry, I'm not sure that.
I think it's best you read the question yourself. So I'm trying to understand directly. So the final fold is very much influenced by non-covalent bonds.
Yeah, definitely. There are those are the one guiding the folding and making the 3D structure. So yeah, definitely they are I won't say the most important, but they are equally important as the covalent ones also. Because without the covalent, you don't have the chain.
Yeah.
And so here we will have a look at the new residue. So this is the arginine residue now coming at that position 188. So the nice thing most of the time is to do a comparative analysis of what happened. So you have this new arginine residues, and it's around this there are some other residues, but the cool thing is that I'm actually not showing that. Hopefully I won't break something, but let me just try this. So the cool thing will be really to have the partner next to it. Hopefully it's not gonna break everything. Yeah, cool. This is what we want. So the cool thing really is this bit here. So you have the arginine residue, and you have the other interacting. So as you can see, there is it's come close to another histidine ring on the other part, and usually, I mean, when things get very close like that, there is a clash, and it's not at the ideal bond, non-covalent bond distance. So, but again, we are not doing anything super advanced here. So it's just basic rotamer search on the mutated residue. And sometimes when after the mutation, what will happen is that actually it's not just this residue that will change coordinates, it's actually the entire environment that will change coordinates. So according to you need to do a more advanced repacking of the entire region here, so that this kind of thing can be solved. But for now, yeah, it is what it is. And now let's just have a quick look at the difference between the surrounding residue between the histidine one and now the arginine one. So we will have a look at here. This is after the mutation. We have the arginine residue. So we have the position 188 on chain B. Yeah. So before, after the mutation, these are the set of residues that we have. Now, before, these were the set of residue that we had. And it's, I mean, as you can see, there are some changes. So for example, this was in close proximity with the histidine. Now it doesn't seem to be the case anymore, position 31. And you have some water that get lost, some other residues might get lost, and some new residue might come here that are not here. So the cool thing is to see the difference between those kind of lost and gain interaction, and depending on that, you will have either a stabilizing effect on your protein structure or something destabilizing your protein structure. And so the best way to do that is actually to look at the energies, and you compare the energy values before and after the mutation. So we have the two structure, the mutated structure, and we have the wild type, which is the original protein. So I'm not gonna run this again because I already did that. But if you are using a notebook, you will have to install this tool called EvoEF. And basically what EvoEF will do is to compute the energy for the protein. So it does two things. I mean, it might be doing other things also, but here we are mostly interested in looking at the stability of the protein. So as the stability change between your wild type and your new protein, the variant. And also we are interested in the binding energy. So if we go back to the structure here, so the binding energy will be what is the chemical energy gained by the interactions happening here. So by changing the residue, obviously we're gonna change that energy value, and that will give us what is the change happening basically. So here we will compute the stability, so between the wild type and our variant. I think should be fine. So in EVF, I mean, you can, if you're interested, you can read about it later. But basically, with the energy values, the lower it is, the better it is. And what we can see here is that we have the total energy for the variant protein is around 305. And we have the total energy for the wild type. So what happened due to that mutation is that we have, even you see the increasing value here, but it is a destabilizing effect on the entire protein structure. So basically, the variant is not that as good as structure as the wild type. So meaning that that mutation is not really stabilizing the protein. The next thing is to look at the binding energy as the interface. So again, we compare our wild type to the variant, and the lower the energy, the better it is. So here we are -27, here we are -12. So we can see that again, even at the interface, the energy there got lowered, which is not that good anymore. And due to all those things, now we have, I mean, this is at a very local level, which cause all the, I mean, butterfly effects leading to a disease person, basically. So small change happening inside the protein, just changing. I mean, actually behind the scene, it's just a change in the DNA. But I mean, as a result, we have a change in the structure, and this complex is not really fulfilling its function that much anymore. And this is the kind of thing that the DeepChain app actually is doing. And I mean, we don't give you code there. You have like an app designed, and you can run your stuff. You can run it from multiple mutations. You can try to see if you try a different mutation, whether you're gonna gain in binding energy, and that's actually protein design. So you can see which mutation is more favorable. You can also mutate different positions or do multiple mutations at the same time. So there are different things to explore. But yeah, just as a quick demo. Yeah.
That was great, Bakri. Just to wrap up on the missense mutation of the nucleotide, is it right that it's just a single nucleotide switch between histatin and arginine? I don't know.
I haven't read the detail of this mutation, but sometimes it's just a single variation inside the DNA. So there is just a change in the base.
Great. And yeah, I just wanted to conclude, and please drop questions if there are questions on this section, but before we jump over to the DeepChain apps side. But yeah, I just wanted to conclude that, yeah, there are lots of amazing tools out here, as Bakri has been showing. So everything is open source, and you can reuse these bits of code. They're not InstaDeep code or anything. We just wanted to give you a handle on how you might analyze the structure of a protein to then extend that for other things you might be interested in if you wanted to explore further in ML applied to protein design, and hopefully just give a feeling that we do have a very nice platform in DeepChain that does lower the barrier to entry to exploring much more complex tasks quickly on top of this code with our own libraries and things. But unfortunately, at the moment, it's not open access to get this level of complexity. There's a small license fee, so we wanted to give some code that would allow people to explore this to start with.
Yeah, I think I'm gonna hand over to Khalil if there is any question. If not, yeah,
I didn't see any questions in the chat, new ones, I don't think, for now. Great. Okay. And so let's jump over to Khalil. Khalil, do you want to take the screen share, or do you want Bakri to keep sharing?
Can you hear me?
I can hear you, yes.
Yeah, we can hear you.
Yeah, okay, I'll try to share from my side.
Fantastic. So Halil is going to move now from the structure-based analysis that we saw with physical model scoring at the end as well, with Evo EF, and a bit more intuition as to what's going on in when you perform a mutation in the protein and this loss of a function, back to these sequence-based language models and what we're showing in terms of likelihood scores of the mutations based on corpora of proteins, and well, how these have been constructed and how you have access to them through DeepChain apps, and the possibility to kind of extend those with the DeepChain apps platform. Go ahead, Halil. Sorry.
Yeah. Yes, thanks. First, so yeah, I'm Khalil, a research engineer at InstaDeep. So I work with the DeepChain team. And yeah, I think we will get a brief, let's say, overview over the biotransformers, which is an open source package by InstaDeep. It's some sort of wrappers over the protein language models, such as PortBERT and DSM, and actually you can see. I will share with you the link. And after that, you can easily use these models and try to include them in DeepChain apps, and contribute to DeepChain apps easily. So yeah, protein language models is actually you can say it's just from analogy to the NLP. It's language model trained on the sequence of the protein instead of just text. So seeing here the architecture of BERT, this is the vanilla BERT. Like forgetting the sentence input, then tokenize it, and yeah, and for the masked language modeling training, also, which is called also in their paper, the self-supervised learning, when the model tried to mask some certain places, which is I think it's 15% randomly, choose these position and mask them, and the model will try to predict these tokens, and based on that, and based also on the result, it will update the weights and improve eventually the representation of the words and the embedding of the of the models. So by this analogy from the text, you see, like you get you just add as input the sequence of protein, and then it will mask some randomly the 15% of the sequence input, and try to, to perform the same analogy for the training, which is the masked language modeling. And so yeah, for that I will try to use biotransformer package, like I said, which is an open source wrapper over these protein models given by InstaDeep. Yeah, and first you should check that you have the GPU in your runtime. So let's, for example, pick the PortBERT model and use it, and use this pre-trained model. So what we are going to do is just getting the likelihood score of, yeah, sorry, didn't install the package. Yeah, so what we are going to do first, we'll see the embeddings eventually of the sequence, which is I think the chain B that you saw with Oliver and Bakary of the 3MZG structure. Yeah, we'll see the first embedding representing the CLC, the CLS, sorry, which is a very common embedding used for classification. Also, we can do the mean embedding of the layers of that of the model, and we can easily also access the probabilities of that the model gives to these amino acids. I mean, for every position in the sequence, you can see that the model try to give a probability for each 20 amino acids, and you can see these probability range. And finally, you can score the log likelihood of that sequence, and it can give us the let's say how this sequence is more probable or how is more close to nature. And you can easily, for example, compare the sequence before and after the mutation and see how the log likelihood, I mean, how the value is, and as the value is close to zero, it will be more stable or more natural, and our mutation will be more preferable. So yeah, I think we still need to wait for the installation.
No problem. Yeah, I think if we can just wait till the last models are installed from biotransformers. If there are any questions anyone wants to go off mute and ask at this stage, we'll just wait for that to load and take a little pause, because yeah, I mean, there's lots of information as well. On my side, I'm just keen that it seems, based on the questions, you do have access to a version of the Colab as well.
Yeah. So yeah, I think it will just download the checkpoint from the biotransformers. And
so all of this that's yeah, you were running through in code, you have access to directly in the playground interface in DeepChain. Should you I don't know if based on the audience, if you just wanted to look at something quickly and preferred the interface of selecting an ID and then seeing the mutations across the structure, that's there and what we showed in the playground.
yeah. So yeah, I think then just run these over our sequence. So for example, if you want to see the embeddings, yeah, you can easily access that. And like I said, this embeddings will be used for your specific task if it is classification task or prediction, like we use that for the NLP. And given a sequence of protein and the data that you have, you can train your model to classify something or predict something. And for the probabilities, so yeah, we are focusing on that 188 position. And you can see that the model give you 20 probabilities for that position specifically, for the I mean for every amino acids, and you can look over these probabilities and see what's the more preferable one that could be, I mean, the
Do you want to just show H the native and R the mutation from our example in the list, Ellie? Can you see that?
Yeah,
I'll
do that. Yes, yeah. For example, yeah, we'll try to do that mutation. So yeah, to see the probability of that position, let me just see. For example, which is in our case in the native sequence, it's I think it's H.
Yeah.
No, it's H. What we want to mate it to is the arginine, right? Yeah. So yeah, you see, so you can, I mean, you can see the probability for the arginine, and definitely you can see the max. What is the maximum probabilities? Maybe like selecting, like you said in the DeepChain platform, selecting the top 10 or top whatever probabilities, and try to play with that. So for the likelihood scoring, this is scoring the, I mean, we scored our native sequence, and you can see yeah, you can see the value. Now we want to try to mutate the sequence in that position, which is estherin, and you want to replace it or to mutate it to the arginine in that position. And after that, maybe like try to score and see the likelihood of that new sequence or the mutated sequence and see the result. So yeah, you see that we have our result is more closer to 0, which is, let's say, more favorable for this sequence to happen. And yeah, so basically,
which goes against what Bakary was showing, right? So in this case, the data likelihood from the machine learning model contradicts the physics model.
Yeah, just let's say likelihood. From the language model, which is not 100% something. So yeah. I don't know if I still have a question, but like I said, you can access the DeepChain apps repo and see. Yeah, it's public repo. You can also read read the description and documentation and how you can implement your app, and also how you can contribute to DeepChain Hub. Yeah. So yeah, I think that's it. I don't know if I have any question.
So in the questions, Abraham has a question on the biotransformers package. You couldn't find it in PyPy.
No, it's yeah. I think the one that you want to install is bio slash transformers.
Yeah.
You can see. It's in the notebook, Brian.
I think that probably
it's in the pipe. It's open source.
Yeah. Yeah, so just as we wait for if there are any other questions, I hope that kind of concludes a loop of some of the things that are accessible to you as an audience within DeepChain and in protein design, maybe inspiring you to think about the domain a bit more, just understand a bit more how pathologies might be built up from the protein level, and how AI could have big impacts in, I don't know, therapeutics, improving our understanding of these problems across other industries that require more insight into the manufacture of proteins as well, and improving their function.
Awesome, thanks guys. I think there was a lot of information.
Yeah, sorry.
There was a lot of information.
Reduce it down, but yeah,
no,
we
but I
gave you a
think that
lot.
was great. No, you definitely gave us a lot of stuff to actually go and look at, which is great. I think that's the purpose of these sort of sessions, right? So I really appreciate all the open source stuff that's available. Just a question on my side. I think it was the beginning of this year. There was an announcement about you guys being able to, or utilizing DeepChain to actually go about picking up an early warning system for SARS and for SARS-CoV-2.
Yeah.
Could you maybe give us more detail around that, like how?
Yeah, so on top of the press release and stuff, I mean, this is work that should be published. There is a bioarchive paper on it, but it's work we've been doing with a client on essentially identifying likely mutations in the COVID virus. So initially around the spike protein that could improve its ability to, for example, evade our immune response. So evade the immunity of the population through vaccinations, etc. And which mutations in the SARS virus on the spike protein could allow it to escape what we considered as kind of general immunity, and so increase its ability to be transmitted. So we saw that a lot with the new variants that we were able to identify with well, for example, Omicron. So actually the combination of looking across language models and 3D structure-based scoring, we were able to combine to look at the sequences of the virus and predict how fit these sequences would be to be transmitted. So yeah, we used a combination of lots of these things we've been talking about to hopefully identify before we have large numbers circulating variants of concern. And that specifically a variant of concern is mostly due to its ability from the fundamentals to avoid things like our immune system, so disrupt the interaction between antibodies and the virus, as an example. So mutations that lead it to escape our immune system and maybe lead to some advantage in its function as well, so infecting us.
It's really exciting stuff, because I can only imagine what the next use cases are coming from here. So protein synthesis is obviously something that you guys are really getting into, but is there any other areas of biology where you can start leveraging off the same sort of modeling techniques as well, over and above protein synthesis?
We are certainly looking across the spectrum, from starting to look more at chemistry and small molecule design. Yeah, so and drug discovery. Right now, we focused on these larger protein-protein interactions, but leveraging the techniques across the pharmaceutical industry, kind of going down to small molecules is certainly of interest as well. Right now, DeepChain is this protein-protein interaction platform, and is really a nice way actually to share the work and educate people on these problems. And that's where we're going with DeepChain right now. But yeah, we are working in projects across the spectrum as well.
Awesome. Some good stuff. Cool. Abraham, you have a question?
So I do have lots of my colleagues on the call as well. So if anyone wants to jump in on any of the answers to these as well, please do. But yeah, any questions from others? Great. So Abraham, you said, mentioning something about processing proteins with 3D convolution. Toma, I guess this was perhaps Toma is one of my colleagues. Yeah, so we certainly continue to look at machine learning methods to replace the physical scoring methods that we've talked about. So whether these are convolutions across the structures that can start to learn things like the stability and binding energies. There is a lot of work, yeah, because these models are computationally expensive. So the reason DeepChain comes with a bit of a license fee for people doing this stuff at scale is, yeah, the compute behind the models is expensive. And if we can lower the barriers to these models, we can start giving you things like 3D based scoring for free in the platform, etc. So there is a lot that we'd like to publish and release more on the ML side, now that we, of course, the physics side is not a solved problem as well. We have these models scoring proteins, and they do quite well. We're doing lots of development in the team on these physical models, but they're not perfect predictions of how the proteins behave in the wild. So in silico design is not a solved problem, evidently, but we're getting a lot of use out of these models, especially in our AI guided design tasks, and transitioning them and continuing to improve on how we can use them for useful projects.
Awesome. Cool. Are there any other questions? I don't think so. So thank you so much, the InstaDeep team, for joining us today. I think that was very valuable information. I've actually been playing around in the background with DeepChain stuff while you guys were demoing. So I think you've really enlightened us with quite a bit of information that would be quite helpful. So next is actually a tutorial with myself around PySwift specifically. We are a little bit over time. So what we've decided to do is actually to separate that out into a separate session. However, if there are any questions, I will stay on for a little bit just to kind of go through some of the basics. But with regards to the actual content and demo, we'll separate that out into another session that we host on our Thursday sessions usually. All right, so let's take a quick 10-15 minute break, and then we can join back on here, or you feel free to stay on here and ask any questions that you'd like to ask.
Fantastic. Thanks, Hachi. Sorry, after the break, what will be the program?
So I'll be doing a tutorial on PySwift.
Oh, okay, yeah, it's yeah.
Okay,
sorry,
awesome.
I misunderstood. I thought you were going to reschedule it, but
No,
it will be
I'll
after 10
go.
minutes. Okay.
Yeah. So I'm not going to do the full demo. I don't think I have enough time for that.
Apologies
But,
on
no problem. I think we're being very ambitious trying to squeeze in all the tutorials, to be honest.
Okay.
But yeah, all good.
Great.
All good. So I'll go through a brief version of it. Have you heard of PySwift before, Oliver?
I have not, only in the context of seeing it on the page, so look forward to learning more.
Okay, awesome. Cool. So I'll join you guys just now. In 10 minutes time, we can start up again. But in the meantime, if you guys want to chat amongst each other, feel free to do so. Are any of you at the Indaba at the moment? Was everyone away from Tunisia?
I'm here, Archie. I just heard you chat. Yeah. How are people going to rejoin?
I'm actually not sure. Probably not, but it's all recorded, so it's totally fine. All right, everyone. I am going to kick off. I actually can't see anything happening in the chat, so if there are any questions, feel free to unmute and just ask me directly. This isn't actually the full demo. I'm just going through the content and just giving you a little bit of a precursor to an actual demo that I'll do perhaps at a later stage. So this is very much a basic entry level that I'll go through, and then we'll go through the actual demo in a future workshop. Cool. All right. So what I'll be talking about today is federated learning leveraging off PySwift. So just some background behind this. The reason how I got into this in the first place was there was a very obvious problem that I found, at least within the spaces that I've worked in, where it was very difficult to share data amongst multiple entities, especially entities that have high levels of privacy and security. For example, the healthcare industry, where it's extremely difficult to share and leverage off patient records, as an example. So how I went about looking for federated learning and how I think it could be quite valuable within the space and solving this problem is federated learning typically reverses the way in which traditional machine learning works. So what you actually have to do typically in the machine learning space, as you might all know, is you have to take your data and you have to centralize data into a location, which may or may not be governed by all the rules and regulations that you need to adhere to. a lot of the time, it is actually governed with all the regulatory needs. But however, especially when it comes to patient records, it's actually quite sensitive levels of information, and it's not enough to have just regulatory encryption standards that are governing that sort of data. So what federated learning does in specific is it actually distributes the way in which you train your machine learning model by allowing each entity to host and manage their own data sets with all the necessary compliance and regulatory measures that need to happen within their individual entities. So for example, within hospitals, so hospitals can retain their own data sets. Example could be things like your patient health records in hospital A, and you can actually retain those patient records within that particular hospital without having to centralize it into a database or something in order to train your models going forward. And the idea is, instead of moving the data into a central location and training your models in that central location, what you do is you actually just pass along your machine learning model from one entity to another. And this PySwift is a framework that allows you to actually follow that methodology. So in this, or at least in this tutorial per se, we're not going to be obviously working with hospital data, but part of my research that I'm doing currently is actually implementing PySwift along with a few other technologies to utilize this within the hospital networks. We've already had quite a few successes with some of the hospitals where we were able to pick out cancer anomalies between 3 different hospitals without having to share patient records. So what actually ended up happening was, because we were able to allow patient records to be retained within their individual hospitals and not have to rely on centralizing those data sets, we were able to get far richer data sets as a result of allowing the privacy and security means to be retained. And we were able to still extract the intelligence in order to further extract the anomalies. So that is essentially how I stumbled upon federated learning. Now, I do have to mention that PySwift on its own will not necessarily allow you to implement federated learning to its full extent. You may need to add other technologies above this. I know this because I've had to, while implementing it, I did come across a couple of issues. However, for more simpler use cases, for example, between edge devices, you can actually leverage off PySwift quite nicely. So yeah, without further ado, let me just walk you through this tutorial that I have on the screen. So this is actually publicly available on OpenMind's website. I will share the link on this chat. Yeah, let me actually share that quickly. A sec. So you can also take a look at it while I. Cool. So this is just an example of what you can potentially do. So in this particular case, when I do finally get a chance to do the demo, what we are going to do is we're going to leverage off multiple devices. It could be a cell phone, it could be a browser-based web service. It could be any sort of edge device that you're leveraging off. And what we're going to do is we're going to pass a model between the different edge devices, and then eventually we will aggregate all of those models into one unified model at the end of the day, without actually having to pass the data between each of the devices. Now, you might be wondering, how is this different from decentralized computation, right? So the idea is, if you look at this diagram, each of these devices are distributed, and you can actually just, if you really want to, you could probably put this on a network and access each of the individual data nodes separately with all the encryption standards. However, it doesn't necessarily give you the ability to pick and choose specific nodes that have more, I guess, more robust features. Whereas leveraging all federated learning and PySwift in specific, you get a little bit more flexibility as well as a little bit more information around the data sets that you have access to, as well as what PySwift does for you off the bat is it adds a noise factor to each of the models based on your model topology, so that when you are actually aggregating your final model set, it doesn't necessarily just combine them together without any intelligence. It just adds a little bit of noise based on how the distribution is looking like in the overall network. So it does. So that's one benefit of using PySwift to do your federated learning instead of a decentralized model. So yeah, I'm not going to go through all of this in here, but the idea, everything I've basically mentioned is pretty much written up in this, written up quite nicely in here. So all you actually need to actually start implementing the solution is you literally just need to download the PySwift model. It's an open source Python library that you just leverage off. From there on, you literally can leverage off any of the Swift nodes that you've created, and from there on, you actually go about building your network. So I'll get into a little bit of detail. So to start off, you obviously install your Swift package. From there, you just basically need to create a there's two types of workers you can create within your network. You have a network socket worker and you have a WebSocket worker. So the idea is each of these individual devices will act as a network socket worker or WebSocket worker, and they will act as your model, I guess your model carrier at the end of the day. So what you would do is at each node, meaning at each device, you create a virtual worker. That's what's happening here. And then for this example, they're just basically creating a tensor that you're going to attach to your virtual worker. From there on, you will try and get information from a specific device. And from there on, you can actually just push that. You can either send that information to the next entity, or you can actually just propagate that up into the final model. So I always prefer looking at a diagram rather than explaining it. So in this case, when we create our tensor node, we send this through to Jake, right? Now, in this case, Jake has now been attached with your tensor that you've passed through. That's the send command here. From there on, you can, when you send the tensor back, so when you get your tensor to get the information from Jake, your Jake worker will be empty, because once you've sent it through and Jake has received it. But once the tensor asks for the same set of information back, it received it. And this kind of acts as how you actually receive your final aggregated model. From there on, you can do a similar thing where you can now that you've kind of aggregated your tensor network, you can pass it through to Jake as well as John. And in that case, it will still be pointing to your initial and your final trained model, but it'll only point and won't necessarily contain the actual information. So that's what's depicted here in this diagram. Any questions so far?
No questions on my side.
Cool. Awesome. Thank you. Great. So similarly, if you would just like to look at each of your different devices and you just want to clear the objects, all you need to do is you just take your workers, so Jake and John, and you just clear the objects. And by the end of it, there will be no models present in your edge devices. So you have the ability to send your model, you have the ability to aggregate your model, and you have the ability to clear your model at the end of the day for all the devices. Cool. Yeah, and once you, so I've explained the send, the get, and the clear, but what you can also do is in certain instances, you may find that you don't necessarily want the mother model to be retained in a specific you don't want the actual final state to be residing in a specific location. So in that case, all you need to do is you just need to move it to a location which you believe the mother model can reside in. So in this case, by using the move command, you can move the tensor that initially resided in Jake's location to John. So simply put, this is essentially how the how federated learning is actually implemented. Yeah, so it's I know it may seem a little bit overwhelming at the moment, but once you do get to play around with the actual package, it does make a lot more sense. Cool. So I'm going to just run through one more very important concept. There's a lot more concepts within the federated learning network that allows you to actually go about implementing it in an actual federated learning device network. But something that is also quite crucial is, just simply passing or just simply storing your data in the different devices and training it independently is not enough to ensure that the model is in fact still secure for training going forward. So what you do also need to do as part of this is add an encryption state, which is called the additive secret sharing command. And essentially what you would do is, very simply, all you would do is just you have an encryption key that you would also pass along with your model at the end of the day. So I know it looks very overwhelming, but that's actually all this is doing. It's just adding an encryption key to the model that you're passing along. So essentially in this example, by adding a encryption key to your entire network, so each of these different arrows that you're seeing here, you're passing the tensor in a more secure space. That's all it's doing. Cool. Then, then there's obviously the ability to decrypt. So everyone that is sitting on the network for your federated learning model also should have the ability to decrypt, meaning that the key can only be utilized by entities that share the network. So it's a little bit more complex, but the idea is when you are decrypting, when you are decrypting your encryption keys, you need to have access to the network. So this is often a problem that I've seen. When your model is being trained offline, you may not necessarily have access to the keys that you need to decrypt your model. So ideally what you'd want to do is you would want to have access to the network in at least a local network connection. So you're on the same local network, and then you wouldn't run into this issue. So just keep that in mind. So yeah, then idea is from there on, once you've got an encryption key that you've implemented up here. So in this case, what all you're doing is you're implementing your encryption key, and then that is added to your network. From there on, every single model touch point would be able to decrypt this key by leveraging off the key that is published to that network with the correct permissions. Yeah. Then homomorphic encryption is a similar sort of follows a similar sort of concept. I'm not going to go into a lot of detail around that specifically, but the idea is that with homomorphic encryption, all you would literally need to do is, following the same approach that we followed up here with secret sharing, you would just encrypt your individual decrypted matches as well going forward. So just an extra added level of encryption on top of what you've already done. Cool. Secret sharing is pretty much what I just spoke about, how you go about ensuring that you share the secrets without necessarily passing out any information. So that's what's happening here. And then finally, this is your actual implementation. So what I showed you were quite a few components. One is how you go about passing your, well, firstly, identifying which one will be your mother model, essentially. Secondly, would be to how you go about actually receiving a model, how you go about sending a model, how you go about clearing a model, then how you go about encrypting your model in 2 different techniques, as well as ensuring that your keys are in fact in the same central network that would be utilized to encrypt and decrypt your models that are being passed between each entity. Now, the next step is, once you've understood those concepts, the next steps would be specifically around how to go about doing this within your actual federated learning ecosystem with actual models. So the example that is in this tutorial is just leveraging off MNIST dataset and then kind of implementing it into those 2 workers I showed initially with Jake and John, and basically training it on the data sets that exist on those individual virtual workers. So these are the packages that you would require. This just basically, loads your data sets for the different workers and on the different individual client devices. So in this case, it's just different phones as an example, but the key point here is make sure that you do in fact create Jake and John upfront as your virtual workers. Remember that could be in the form of your network workers or your WebSocket workers. And then from there on, when you do in fact train your data sets, just refer to them within this ecosystem with the relevant packages. Then the next step would be to just go about building a model based on those 2 virtual workers that you've identified. And then you just build the model with those 2 individual client devices. Then you can go about training the model as per normal. The only caveat in this case is, if you can see here, remember, so you've built your models, but that model resides in that main location, right? So if I can just go back to the original diagram, in this example, well, in the code I just showed you, that would essentially be this owner here, right? So you build your model in your owner. And then going forward, you would just need to ensure that you send the data to, you send your model data to the appropriate locations. You would just need to, and the rest of this is pretty much how you go about training your model. Then the final step is you just need to ensure that once all the data has been gathered with the appropriate data locations, you just need to get the final output back into the owner stack. And that's pretty much a full trained model then going forward. Then you just go about testing and evaluating it after the fact. So yeah, that is a very rushed version of leveraging off PySwift. Are there any questions so far?
Yes, I think I reached you late. When you start. So yeah, I have a question about how the server expecting the data format from the users. How you can handle the cases that you are not getting the right data from these devices. Or
I love that question. So it's actually, that's the reason why I mentioned a bit earlier. I did mention that just leveraging off PySwift on its own is not enough, because PySwift just gives you the mechanism to ensure that you have, the ability to pass the data along, get the data, and then kind of aggregate it. So what you do need at these individual client locations is the ability to have a schema registry, right? So at these individual client locations, well, at least this is how I've implemented it, at least, is at the individual client locations, there needs to be a schema registry to which the data adheres to. And how I went about implementing it is in the owner itself, I have versioned it to a specific schema. And then I've passed down that version as part of a parameter within the model send command, so that every data set that is actually passed through to the model does adhere to that schema. If not, then it's rejected. And what happens is a notification is then sent to the client details. So that's how I went about implementing it, so to ensure that the data sets that you do receive is in fact according to the correct standards.
Yeah, great, thanks a lot.
Awesome. Cool. Any other questions?
Yeah, I had a question on just the motivation for people to provide their data. Is it, perhaps I just missed it at the start, is it really the knowledge so they get a better final product, a better final model, the motivation to share this sensitive information.
So that's, sorry, I cut you off there.
No,
My bad, sorry.
I was just trying to work through the use cases. So
Yeah.
I guess people would join the network to get a better final model, but they're not able to distribute their data. Is there some extra incentive for them, like quantifying how much improvement to the model your data set gave, etc., etc.?
Yes, so the particular, I mean, for my research, the reason why I went down this route is for the first obvious motivation is the fact that you don't necessarily want to share your data, and you don't want to centralize your data in an unknown location. You want to have the ability to keep your data sets where they reside, right? That's the first thing. And the reason for that is regulatory compliance, as well as you don't know what other people are doing with your data. So, data sovereignty and all.
Mm-hmm.
The second reason is from a network perspective, it's actually, from a network and from a speed perspective, it's so much more faster to allow people to retain their data instead of having to go through the hard yards of centralizing everything and getting everything to the correct format. Whereas if you're distributing this network, what you're doing is you're rapidly increasing the time to which you get access to all the different data sets as well. So it's kind of twofold essentially. And at the end of the day, what you do have is obviously a better performing model, because you have more data sets and more, more input at the end of the day.
Mm-hmm. Are there any technologies that combine this federated learning then with blockchain and attach some token towards the training that you do at your node, etc.? I imagine there are.
Yes, there is. Okay. Actually, so currently, I don't know if this is public, but Google Keyboard is starting to look at that specifically. Okay. But yeah, it's definitely in the works, and it's pretty exciting space to be in.
Cool.
Awesome. Any other questions? Well, if not, I have a question for those of you still on the call. Having seen what you've seen and having understood the use cases that this could potentially be helpful at, are there any things that come to mind around where you could potentially use this going forward?
That's funny, because that was actually my question for you. So I was wondering, where are the areas of impact for this? Like there are some areas where things have been like not shared so far. So I was just curious about that.
Yeah. Okay. So that's my area of research, right? In healthcare. It's definitely one of the areas where data just does not travel fast enough, and as well as with all the compliance stuff that we generally have to jump through, it's just so much more difficult to get access to data as well as create a representative model of a community. So definitely in the healthcare space, it's quite crucial. I have started, at least with my research, trying to implement this and showing value around it. But yeah, the more people that I can get on board, the better, essentially.
Is there in the finance industry, I don't know, like in the banking industry, probably?
I'm sure there is.
They share the data.
Yeah, so I work in the financial industry as well, and I know especially in the credit risk space and fraud detection space, it could be quite helpful. Yeah, I haven't tried it yet. It hasn't been something that I actually went about implementing it, but I can imagine anywhere where there is some sort of data security issue, or there just isn't enough of a network to connect enough information together, this is probably the best way to go about doing it. Yeah. And it's not the only way, right? There's multiple ways of doing this as well.
I was trying to think on things that relate to what we do, or at least what our clients are interested in. I think this is a bit half-baked as an idea, but often there are structures or drugs that companies might design, though they end up not using for some reason. Perhaps that information could be shared to some machine learning model that looked at likely candidates for a disease, given that pharmaceutical companies have synthesized these compounds, but they've decided not to use them. Perhaps they don't want to share them openly, but I guess a model could be trained on these.
Oh, definitely. I think so, especially with the accidents. That's a good one. So it's not so half-baked, actually. It's actually a very good one.
That is what I can offer and thinking in the kind of space that we're looking at the moment.
That's awesome. Cool. All right, guys. Any other questions, comments, discussions? Let me stop sharing. All right. If not, thanks for joining, everyone. I will make sure the recording is available. We'll probably send it through to those who have presented just to make sure you're happy with it. But thank you so much for joining. And I definitely learned a lot from all the presenters today. Thank you so much.
Thanks, everyone. And thanks on your side as well, Archie. Cheers.
Thank you. All right. Bye.
Bye-bye.
Bye-bye.